Hello everyone and welcome to Pocket Size Bio with Dr. Casco. In today's video we're going to continue chapter 5 of the Campbell biology textbook. So let's get to it. We left off with the lipids, right? So we're going to continue as section 3, which is lipids are a diverse group of hydrophobic molecules. So lipids are not true polymers. They're not big enough to be considered macromolecules, right? There's no intense chain of monomers that is linking up like you see in nucleic acids or in amino acids or in carbohydrates. So they get these type of molecules get grouped together because of several properties, right? They are hydrophobic, meaning that they mix poorly in water. Right, they're based on a molecular structure per se. They consist mostly of hydrocarbon regions. They might have some oxygen associated within the structure. They do have some variety of form and, of course, a great variety of function, as we're going to talk about later. The groups within the lipids include waxes and pigments, fats and phospholipids, and steroids, as we're going to look at each of them in more in detail throughout the section. So we begin with fats, right? These molecules, they're not polymers. Again, like I mentioned before, they are large molecules and they are assembled from smaller parts, particularly glycerol. Okay, so you have, I'm going to go a little bit higher here. So you have a molecule of glycerol, which is an alcohol, and it consists of 3 carbons, and these 3 carbons each have their own hydroxyl group, right? As you can see here, 1, 2, and 3. And this glycerol can bind to another molecule that is called a fatty acid, right? And particularly 3 fatty acids. And these fatty acids are essentially carbon chains, right? Hydrocarbon chains that can range from 16 to 18 carbons, this carbon skeleton, and one end of them has a carboxyl group. So this carboxyl group, when it joins, when it essentially, after undergoing dehydration synthesis, this carboxyl group with this hydroxyl group remove a water molecule and they bind together to create an ester linkage. Okay, you get this happening between all 3 carbons of the glycerol molecule, and then you have your fat molecule. And this fat molecule in itself is called a triglycerol, a triglycerol or triglyceride. Okay, so you have 3 fatty acids within a glycerol molecule. That interaction, and in itself, these type of molecules, if you notice, is just carbon and hydrogen. The great majority is carbon and hydrogen. Because of this, it creates a nonpolar environment and results in a hydrophobic property. Because of this, fats do not mix well with water. So basically like oil and water. Again, as I mentioned before, fatty acids join a glycerol molecule via dehydration reactions, creating an ester linkage, liberating a water molecule. Okay. Talking further about fats, they're subcategorized into saturated fats and unsaturated fats. And this is very common. Maybe you've heard this before in terms of nutrition, right? What does it mean to be saturated? Essentially, the structure does not have double bonds. Okay. So let me just zoom in here. All right. So you have, for example, at room temperature, right, a saturated fat, like the fat in butter, right? The molecules are tightly packed together, and this is because there is no double bonds. So the hydrogen and the carbon skeletons of the fatty acids it's being completely consumed by hydrogens, right? The bond potentials, right? Every carbon has four possibility to have 4 bonds. So these hydrocarbons are filled with hydrogen as much as they can. So they're depicted by these little zigzags here, this hydrocarbon chain. Okay, so because of that, you have these substances that at room temperature, they're generally a solid, as opposed to unsaturated fats that at room temperature, you have these substances that are liquidy, right? Like olive oil. Okay, you don't have as many hydrogens as you can, right? Just because there's double bonds present within the fatty acid structure. Okay, so if you see this double bond, and a result of these double bonds is that you get certain bends, right, within the structure, and they're referred to as kinks in terms of the hydrocarbon chain. Okay, so these are unsaturated fat molecules, and these are the ones that you refer to as oils, right? And generally, they have the double bond forming a cis double bond, which causes the bending. Okay, so just to go back to my notes here, okay, nearly every double bond that you see in these unsaturated fats is a cis double bond, which creates a kink, right, in the hydrocarbon chains. Some examples of fats that are saturated is lard and butter. Okay, so these are solids at room temperature. It is possible to see hydrogenated vegetable oil, right, in the supermarket when you go shopping, where you have to have these labels, that are unsaturated fats that have been converted to saturated fats, such as margarine and peanut butter. Okay, and continue on to the next page here. All right, a little bit of medical related information here. Diets that are rich in saturated fats, they contribute, or they could contribute, they do contribute, right, to cardiovascular diseases like atherosclerosis. And what happens in this particular disease is that you have deposits that are called plaques, right? They develop in the walls of the of blood vessels, right? And that's a big problem, because if you're having plaque, these fat deposits that are accumulating in the walls, that's going to inhibit or greatly decrease the efficiency of blood flow, ultimately to the point that you're getting a obstruction, you're reducing vessel integrity, you're reducing blood flow, essentially creating clots. And then, of course, if blood isn't flowing effectively, you're not getting enough blood to where it needs to go. Okay, very, very important, very serious disease, right, related to health. Hydrogenating vegetable oils also produces trans double bonds, also, right, rather than unsaturated bonds. Trans fats, very important, can contribute to other health diseases like coronary heart disease. Okay, and this is very common to baked goods and processed foods. So it's very, I don't want to say it's very unhealthy, but you have to take care, right, of what you're eating. FDA requires labels of trans fats in food products. Functions of fats, right? As we all probably know, right? One of the main functions of fat is energy storage, right? Hydrocarbon chains are similar to gasoline molecules, right? And a gram of fat stores more than 2 times as much energy as polysaccharides, sugars like starch. So it's a good source of energy. Vegetable oils stored in seeds, they store energy compactly in seeds, right? Animals, being organisms that move around, they contain their energy in fat. More specifically, the fat that we have, we contain it in very specialized cells that are referred to as adipose cells, which can enlarge and shrink depending on how much fat you actually have in your body. Okay, so this adipose tissue can also serve for organs as cushions or as a source of insulation for certain organisms like whales and seals, depending on the environment that they're living in. Okay, and depending, on how much fat, you can see it more evident in the body. There's different areas in which fat could be stored, right? In around your thigh area, under the heart area, right, on the belly, and so forth, right? And there are different types of fats, as we'll see in the later sections. Okay, so fats, phospholipids, another group of lipids, very, very important group of molecules. Of course, cells will not exist without these particular group, these lipids, right? They're the major constituent of the cell membrane. Okay, you do need to know the structure of phospholipids. Essentially, you have two fatty acids attached to a glycerol molecule. Okay. The third carbon within the glycerol is a phosphate, is attached to a phosphate group, which is an additional charged molecule. So that gives it very special properties that we're going to take a look at. Okay, so you have, taking a closer look at the diagram here, you have your glycerol, okay, structure, your glycerol molecule here, and you have that those three carbon chains, and those three carbons, and then two of these carbons you have fatty acids, right? These two fatty acids, and then you do have a double bond kink there within the tail region that they call, right, the hydrophobic tail region. The hydrophilic head, coming from this choline and phosphate group, creates that little circular head that we all think about as the phospholipid. And this is a very standard annotation for phospholipid as a symbol, right? That you're going to be seeing this throughout the text, right? So you have a phosphate group, as I mentioned before, which is negatively charged. Choline, right, can also be attached to the phosphate group. All right. So you have a hydrophobic tail. Hydrophobic, of course, it's saturated with hydrogens, right? So it's excluded from water, or it avoids water. And then you have your hydrophilic head, which has an affinity for water. So because of this, we're going to see this in the later chapters, phospholipids within biological structures creates a phospholipid bilayer. Right, and it can spontaneously self-assemble into a double-layered sheet, which we refer to as a bilayer. Okay, and it helps protect the fatty acid part to be away from water. All right. So at the cell surface, the phospholipids are arranged in a bilayer form, every cell. Okay, so that creates, since you have this barrier here, you have an external environment and an internal environment, right? It creates essentially the cytoplasm and all the organelles to stay within the cell, and it separates the external environment to have a distinct region between the outside and inside. So essentially it creates a boundary. All right. Continuing on with the steroids, right? So steroids are essentially lipids that are characterized by having four fused rings, as you can see in this picture here. And there's different types of steroids, and they're distinguished by the chemical groups that are attached to these particular rings. A very common one that we're going to take a look at more in detail is cholesterol. Okay, it's a common component within cell membranes, and it's embedded within the hydrophobic area of the bilayer. And this kind of cholesterol molecule can serve as a precursor to produce other steroids, right? Like sex hormones. Steroids generally made in the liver through our diet also. And high cholesterol, we're going to see this also later, can contribute to diseases like atherosclerosis. Okay. Just a little bit of questions there for you guys to take a look at in your own time. So we're going to jump into the proteins now. Okay. All right, so 5.4. Proteins include a diversity of structures resulting in a wide range of functions. Just to put some perspective, so nearly every dynamic function of a living being depends on proteins. Proteins coming from proteose, meaning first or primary. And so proteins account for more than 50% of the dry mass of most cells. So it's fundamental. This whole group, right, of molecules is so fundamental to almost everything organisms do, and it kind of governs how our dynamics, our molecular dynamics works. Right, so wide variety of functions, right? Before I get into the possible functions, life will not be possible without proteins. In particular, one group of proteins, which are the enzymes, right? Enzymatic proteins, they are a particular group that helps regulate metabolic activities because they act as biological catalysts, meaning that they can help speed up chemical reactions, right? Without themselves being consumed into the chemical reaction and the products. That means they can keep being used over and over again. Okay, so again, this is something we're going to get into a little bit later. So you have several groups. You have enzymatic proteins, you have defense proteins, transport and storage proteins, hormones that can regulate biological activity, receptor proteins, structure and contractile, right? So as I mentioned before, enzymatic proteins help kind of regulate chemical reactions, help accelerate, moreover, chemical reactions, break down and create molecules. Defense proteins, basically antibodies, they help inactivate and destroy foreign invaders like viruses and bacteria. You have storage proteins that help, they're a major source of amino acids for mammals, for baby mammals, right? Ovalbumin, okay, you have this within the eggs, a good source of amino acids. Transport proteins, we're going to see, especially with the cell membrane, you have carrier proteins and ion channels that help chemicals and molecules go, in a particular direction within the cell membrane. Hormone proteins help regulate certain processes, organism activities like insulin, right, which helps regulate glucose, right, and blood sugar concentrations. Receptor proteins, these are very special types of proteins that help detect a chemical stimuli, right? They can be found within the cell membrane, right, to receive a molecular signal, to relay some signal into the cell or out of the cell. Contractile and motor proteins, these are more for movement, right? Basically muscle. Actin and myosin are very famous proteins there for the contraction of muscles. Structural proteins like keratin, right? Collagen. These are proteins that can be found. Keratin can be found in hair, horns, feathers, skin appendages, right? Silk fibers, right? For insects and spiders. Collagen and elastin also, right, are another protein type that are very important for providing, connective tissue, a framework for connective tissue. So wide variety of functions. And as we go along through the book, you're going to just inevitably remember many of these protein structures, okay? But even though there's a good variety of proteins, they and they have a unique 3D shape, they have a unique amino acid sequence, right? They're essentially made up of 20 a set of 20 amino acids. Depending on the sequence, you can have a repetition, a use of more of one type of amino acid than the other, depending on the particular sequence, right? And these amino acids are essentially linked together by peptide bonds. And when you have amino acids chained up, you have a polypeptide. It's a polymer of amino acids. They're referred to as polypeptides. And depending on the sequence, which type of amino acids you use, that will determine the function of the molecule. That will determine the 3D shape of the protein. Okay, so it's very, very important. Sequence is king when it comes to amino acids. And of course, it comes from DNA, right? The sequence of DNA. There's a little bit more caveats in there, though. So you have a very specific 3D shape. That 3D shape will give you the particular function. Okay. So focusing a little bit about the nature of amino acids, right? The monomers, they do share some commonalities amongst each other. Okay, they have an amino group. As you can see here, there's an amino group where you have a nitrogen with 2 hydrogens. You have a carboxyl group, okay, stemming from that carboxylic acid, thus the name amino acid. Okay, you have a central carbon, is referred to as the alpha carbon, a hydrogen atom on one other bond of the carbon, and you have a R group, right? An R side chain group. So this R side chain is what gives and what differs between one amino acid and another, and it gives it its particular characteristics, right? So you can replace this R group with several things, as we're going to see. It could be another hydrogen here. It could be a hydrocarbon chain or other functional groups. So the side chain will determine the physical and chemical properties of the particular amino acid, okay? The R group. You can fill it in with X, okay? So let me just show you some examples. Okay, so there's 20 type of amino acids. Now, depending on what's bonded to this particular site, you have particular properties, right? So these R groups that essentially have a non-polar side chain, okay, like a hydrogen, CH3, right, and so forth, they're all grouped together as hydrophobic. Okay, and you have glycine, alanine, valine, leucine, isoleucine, and so forth. Generally, they're non-polar molecules. You have another group basically being hydrophilic, serine, threonine, cysteine, tyrosine, and so forth, that have sulfur, oxygen atoms that are coming in and creating a polar side chain. So they're hydrophilic. Okay, and then you can have hydrophilic in terms of being acidic or basic, that are positively charged or negatively charged. Then again, can give a particular property to the amino acid and the polypeptide in general. Okay, so every group, every amino acid has this particular property, and depending on similarities, they're grouped into particular groups, right? So we are going to take a look furthermore. So talking about amino acids, that's going a little bit more detail with polypeptides, a chain of amino acids, of different types of amino acids. 2 amino acids can be joined together by dehydration reactions. Again, so you have a carboxyl group and an amino group reacting together, losing a water molecule. That water molecule being lost will enable them to join together, creating a peptide bond. Right? So lipids have ester bonds with the fatty acids and glycerol. Here you have a peptide bond. Okay, so these peptide bonds will repeat with every amino acid that comes in, right? So it essentially makes a backbone. And that backbone will enable you to chain up all the amino acids until you're done with the sequence that is predetermined by the DNA and RNA, right? So One thing that's interesting is that the amino acids, as they chain up with these in this via these peptide bonds, the R groups will always be extending outward of the backbone. Okay, they will be exposed for actual use. All right. So polypeptides, they can range from a few to 1,000 plus amino acids. Okay, many amino acids. And you have a unique sequence of amino acids, again, predetermined by mRNA and DNA and so forth. One end is referred to as the N-terminus, and the other end as the C-terminus, basically the amino side, and then you have the carboxyl N side. Okay, so you have these free functional groups there. Again, very important to note, for you to know, the chemical nature of the molecule is determined by the types of amino acids that are being used and the sequence in which you have these amino acids, and more particularly, the sequence of the side chains. All right, so talking about that more in detail, protein structure and function, right? I do want to mention, and students do get confused with this, that polypeptide is not the same thing as a protein, okay? The term polypeptide is not synonymous to protein. It's like saying having a yarn versus a sweater that is actually made, sewn up, and ready for use, as opposed to just throwing you a chunk of yarn, okay? So it's not ready to be in its optimal form yet, okay? So a functional protein is a structure arising from one or more polypeptides that are precisely twisted and folded and coiled into a very unique and precise 3D structure, okay? The amino acid sequence of each polypeptide will determine the 3D structure, therefore the function, okay? So after polypeptide synthesis, the chain may fold spontaneously and assume the function there, okay? Different parts can fold within itself, and there's different types of folding, of coiling, as we're going to take a look at. So depending on the sequence of amino acids, you're going to have either globular structures, as you can see here. This is a very globular type of structure, or very thread-like fibrous structure. So you have globular structures and fibrous structures there. Okay? Functions of protein usually is to recognize and bind to some other molecule. As you can see here, here's an example of an antibody. So here's your particular protein, and it's binding to some other protein from, let's say, a flu virus. And that binding is very complementary. Okay? So because of this structure, you get a function, and that becomes an emergent property, as we talked about before. I do want to mention here, it's very interesting how that complementary, that binding capability, we can see that particular area of this antibody protein fits nicely within the flu virus protein, right? Basically nullifying the virus. All right. So what you're seeing here is just a 3D model, a computational model of how proteins and amino acid sequences and the overall 3D structure is shown as a computer. It's very important to know this, okay? There's different ways in which proteins can be visually represented. You have a space-filling model that emphasizes on the overall globular shape, okay? And each little kind of ball represents an atom. You have the ribbon model, this very nice representation of how you can see the coils that we're going to talk about in a bit, the folds and coils that the protein and the polypeptide sequence and overall protein is showing. And then you have a wireframe model. All right, and this basically shows the side chain that is extending from the backbone there, okay? Again, complementary shape to a target that you want to have. Okay, let's move on to the next section here. So in terms of 3D structure, there's 3 levels that we're going to take a look at: primary, secondary, tertiary structure, and then you have an additional quaternary structure that, depending on the protein type. Okay, I will go over this essentially directly from the book because it's so important for you to know how these structures work. It's something fundamental to molecular biology, and especially if you're getting into bioinformatics, you must know these different levels of protein structures. Let's get to it. The primary structure is essentially the linear chain of amino acids. Okay, the book gives this example protein, which is transthyretin. Okay, it's a globular blood protein that transports vitamin A and of the thyroid hormones, and one of the thyroid hormones, right? So this is focusing now on primary structure. This protein is made up of 4 identical polypeptides. So we're going to see how they kind of integrate each other at the end, right? And each polypeptide is composed of 127 amino acids. So here we can see just one of the polypeptides, okay, and that chain of amino acids, 127, is shown here in this little kind of a linear snake appearance. Okay, so when we take a look at these representations, it's very nice, and it's quite standard that you enumerate the amino acids, and the same thing goes with nucleotides as you're kind of showing sequences, right? So you have glycine being the first amino acid, and depending if you're using different software and whatnot, you can change the annotation here, but it's enumerated, right? The 5th amino acid, the 10th, and so forth until you get to the the last amino acid that you're looking at. In this case, 127, which you have here, and then you have your carboxyl end at the beginning. You have the amino end here. Okay, so primary structure, it only talks about the sequence of amino acids. Okay, starting from that first amino acid, glycine, all the way down to the last one, glutamine, if I'm not mistaken. Okay, so primary structure is like the orders of letters in a very long word. Okay, depending on the DNA sequence that will transpose after translation, you will have this particular sequence. We're going to get into this in a lot more detail. Essentially, sequence is king, right? You're going to have your product based on particular amino acid sequence, which is determined by DNA sequences. We'll get into that in the later chapters during protein synthesis. Okay, it is not random. It is determined by, again, inherited genetic information. So by having this specific amino acid sequence, we now jump into the secondary structure, which is regions are stabilized, right, by hydrogen bonds between atoms of the polypeptide backbone. So it's important to note, so this secondary structure is governed by hydrogen bonding. Okay, hydrogen bonding. So most proteins have segments of their polypeptide chains repeatedly coiled or folded. There's 2 types of structures you can have in this secondary structure. You can have something called an alpha helix, which is basically a coiled region. Okay, or you can have the beta pleated sheets. Okay, now what governs this again is hydrogen bonding that is occurring between the oxygen molecules, okay, and the hydrogen molecules, the hydrogen atoms and the oxygen atoms, sorry, which are, have that partial negative charge for oxygen and partial positive charge for hydrogen. Okay, so individually, as we know before, hydrogen bonds are generally weak, but when you have so many of them working together, it starts to create a more fixed structure, a more stable structure. Okay, so again, let's go into the two types of structures that you can have in this level. One of the secondary structures is the alpha helix. So this is a delicate coil, and you have hydrogen bonds occurring every fourth amino acid, as shown above. So roughly, you can see as you're going along in this twisted chain, every 4 amino acids, you're going to have a hydrogen bond occurring between an oxygen atom and hydrogen atom. And this interaction will ultimately develop that helix that we see here. Okay. Now, again, globular proteins, they can have multiple stretches of helixes, right? That could be separated between non-helical regions. So you can have particular areas of helices and then some, loose stringed region, and then it can continue on to another helical region. It doesn't have to be just one entire fixed region of alpha helices. An amino acid sequence can have a lot of alpha helix or beta chain, beta pleated sheets, or a combination of both in some kind of ratio. Okay, so the other secondary structure is the beta pleated sheet. Okay, I'm going to go up in a bit. So 2 or more segments of the polypeptide chain are lying side by side. They're connected by hydrogen bonds between essentially parallel segments. And these segments, because of the hydrogen bonding, they are kind of folded, a little bit higher and lower and higher and lower between different regions. Okay, and you could have orientation where the amino acid sequence could be going in a particular direction, and so forth and so forth. And they're designated by these kind of arrows, as we're going to take a look at a ribbon model of a protein or a polypeptide. So you have your alpha helixes in this ribbon form, and then you have these beta sheets in this kind of arrow flat, this flat arrow form here. Okay, but ultimately you only have hydrogen bonding occurring within this level. All right, and depending on the protein and depending on the function, you're going to have more of one than the other, or a good mix between the two. Okay, beta pleated sheets make up the core of many globular proteins. In the case that we're talking about right now, transthyretin, and dominate some fibrous proteins also, including silk protein of a spider web. Okay, going on to the tertiary structure, the 3-dimensional shape now. The overall stabilization occurs between the side chains. So here's where the R groups, right, are coming into play. Okay, there's interaction occurring between each of these side chains. All right, and here's an example now of this tertiary structure, right, or a computational model here of your polypeptide. We see the beta sheets and the alpha helixes that they're interacting now within regions based on the R groups. Okay, so tertiary structure is the overall shape of the polypeptide, and it results, again, from the R groups, the interactions between the R groups, right, of different amino acids. There is one interaction that is commonly referred to, but it's not really necessarily an interaction. It is called the hydrophobic interaction. And what happens here is that part of the polypeptides that are hydrophobic, or the amino acids regions that are hydrophobic, are going to tend to go into clusters, and they're going to kind of shield themselves into the core of the protein, into the inner parts of the protein, so they could be away from water. Okay, it's an exclusion of nonpolar substances by water molecules. So it's not that they're necessarily interacting, but the repulsion and the interaction of hydrophilic regions are making them go in the core of the protein. Okay, ultimately they're held together also by van der Waals interactions, stabilizing them in these particular areas. All right. So again, you have interactions between R groups. Hydrogen bonds is also occurring between different R groups, right? Whether you have, negatively charged side chains with positively charged side chains, you're going to have some type of interaction here. Okay, as you can see here, some examples. You can have ionic bonding between particular R groups, van der Waals interactions within the hydrophobic groups, or hydrogen bonding just from these kind of partial charges within these different R groups. One particular type of bonding is a disulfide bridge, where you actually have covalent bonding between the sulfur atoms between these two different R groups that have sulfhydryl groups, right? The SHs. So they get bonded together, greatly having creating more stability within that protein. Okay, so these are covalent bonds, as you can see here. All right, so that's tertiary structure, and then you have quaternary structure. Talking again about transthyretin, you have 4 identical polypeptides. They come in and interact together to create the actual protein. So this is the quaternary structure. Okay, it's the overall protein structure that results from the aggregation of these polypeptide subunits. Okay. Another example here is collagen, right? It's a very fibrous protein that it has, it's a very helical polypeptide, and it's intertwined into a larger helix, which gives it a very good kind of tensile strength, right? And of course, it suits collagen because it's collagen is very useful for connective tissue in the skin and bones and tendons and so forth, right? So you can see this interaction here of these kind of helices intertwine amongst each other to even provide even more strength. Okay. Another example is hemoglobin, where you see a quaternary structure. Okay, I'm sure we're all familiar with hemoglobin. It's an oxygen binding protein of the red blood cells. And here you have an example of globular proteins where you have a structure of 4 polypeptide subunits where you have different kinds of subunits. You have 2 alpha subunits and you have 2 beta subunits that are working together ultimately to kind of house and transport oxygen, right? They work together with a heme group, right? A non-polypeptide component called a heme or a heme molecule, and it's these heme molecules that enable the bonding of hydrogen and to the transport hydrogen, right, within the red blood cells. Okay, so this is an example of a globular protein where you have 2 different types of subunits, right, totaling up to 4 different 4 subunits within a molecule. So make sure you understand the different levels of protein structures. Okay, primary structure, amino acid sequence. Secondary structure, you have alpha helixes and beta sheets. They're created by hydrogen bonding. Tertiary structure is the overall shape of the protein, of the polypeptide, based on the R groups, right, of the amino acids. And then you have quaternary structure, where you're having multiple polypeptides working together. Okay, let's continue on. Sickle cell is a very nice example of what happens when you have a normal protein, and then that protein got a mutation on the DNA sequence that creates a change to that protein. Again, talking about structure fits function, an essential concept in biology, right? So sickle cell disease is a change in primary structure. Primary structure, which ultimately will change the function of the molecule. So now we're talking about sickle cell disease, and here we have an example of what happens whenever you change the amino acid sequence of a normal protein. Okay, so what is sickle cell disease? It's an inherited blood disorder. It's caused by a substitution of one amino acid, valine, right, for the normal glutamic acid. All right, so you have your normal glutamic amino acid, right? This is for normal hemoglobin, and you replace it with valine in this sickle cell situation. Okay, so normal red blood cells versus sickle cell red blood cells, there's a big difference in shape, and of course there's in the shape of the actual red blood cell, and also the protein itself, and this has major consequences as we're going to take a look at. So if you see here, the secondary and tertiary structures of hemoglobin, instead of being this nice globular structure where you have the beta and alpha subunits, you have this kind of little sickle appearance of the protein. Now, what happens with this hemoglobin is that it kind of lodges onto other hemoglobin proteins and it creates a chain. It aggregates as a fiber. Okay, and this aggregation of a fiber is kind of stuck on the red blood cell, and it creates this sickle appearance. Okay, it deforms the red blood cell, and again, this will lead to very serious conditions within the patients. Right, they experience crisis periodically, a lot of pain. Red blood cells get lodged onto the blood vessels. They can create clots. Okay, so it's a very serious condition. So again, you're losing the structure, the sequence, which loses the structure, which ultimately loses the function. Okay, but what determines protein structure? You have a polypeptide chain with a very specific amino acid sequence, right? And ultimately that it's arranged in a 3D shape, right? So what influences protein structure could be several factors, right? The folding of the secondary and tertiary sequence, it could be influenced by the environment, right? pH plays a role. It could break down hydrogen bonds, the interaction between the R groups. Salt concentrations can also influence, and of course, temperature can break chemical bonds, making the protein lose its shape. Whenever the protein loses its shape, that is called denaturation. Okay, denaturation. So ultimately, the protein becomes inactive. Protein becomes inactive. It has lost its form, therefore the function. Okay, solvents can also denature proteins depending on the characteristics, right? The hydrophobic solutions, chemical agents can disrupt hydrogen bonds, ionic bonds, and disulfide bonds. So chemical agents also can influence denaturation. Temperature, as I mentioned before, we see this, I'm sure you've experienced this, making some eggs where they turn white. Okay, that's denaturation of proteins in a very everyday situation. Denaturation can also be seen in high fevers, very dangerous to the body. That's why it's so important to regulate body temperature. If the temperature gets too high, enzymes could get denatured, therefore losing the function, normal molecular functions. Okay, so very, very important to regulate temperature in terms of fever. It is possible, though, for some proteins to return to their functional form if you remove it from that naturalizing agent, and or treating it with another solution, right? But by themselves, it's sometimes difficult to get it back to renature, if I can say. Okay. Protein folding within the cell. This is a very, very interesting topic. That the whole idea of how to get the protein to actually fold on itself, right? Biochemists, bioinformaticians, the computational biologists, chemists work together to kind of figure out the amino acid sequence and the structure of over 160 million proteins. But it's very difficult to experimentally know that, right? Because there's many stages to analyzing proteins, and there's many variations of this protein. So this is a good area of research, especially with the coming of AI and AlphaFold. we could get a sense of how to predict, a particular protein. So folding correctly is important to know because we can identify when misfolding occurs for certain diseases, right? Like cystic fibrosis, Alzheimer's disease, Parkinson's disease, and mad cow disease, where you have a particular disorder stemming from misfolded proteins, right? So it's very important to understand how naturally a protein works, right, in its structure. All right. So how do you determine 3D structure? There's several ways, right? One of the most common ways is X-ray crystallography. When you purify a protein, you can create a 3D model based on the refraction of the X-ray or the diffraction of the X-ray. Okay, onto a detector there. Nowadays, there's more bioinformatic approaches to analyze the 3D structures. Again, like I mentioned before, AlphaFold that just recently came out a couple of years ago, which has a big breakthrough in kind of predicting the 3D structure. Another challenge that scientists have is that proteins sometimes don't achieve their actual 3D shape until they interact with another component or another molecule or another type of protein. So it's difficult to really, predict the 3D structure at a particular time. So that's a little bit tricky there. This situation could account for 20 to 30, percent of mammalian proteins. So there's a big undertaking there. Okay. Generally, proteins are intrinsically disordered, so they haven't fulfilled their 3D shape yet. Okay. If you want to know more about the X-ray crystallography, here's a panel to show how it actually works. I will let you take a look. You can pause the video to see how it actually works there. Okay. All right, so we can jump in now to the last part, which is nucleic acids. Nucleic acids, they store, transmit, and help express hereditary information. So again, similar to amino acids, to carbohydrates, you have also nucleic acids that are built from monomers, repeating units. Okay. Primary structure of amino acids determines protein shape. But what determines the primary structure of the amino acid sequence? All of that is enveloped into what we know as genes. So genes are particular sections of DNA. There's another type of macromolecule. I'm sure we've all heard of nucleic acids and DNA and RNA, right? They belong to the group of nucleic acids. And nucleic acids are long macromolecules that are made up of monomers called nucleotides. Okay, nucleic acids, there's two types of nucleic acids: DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. Sorry about that. So ribonucleic acid for RNA. And these molecules enable organisms to reproduce. They're complex components from one generation to the next. So they serve to pass down genetic information from the parent to the offspring, and they function in DNA gene expression, right, to create proteins. So the process of which DNA directs RNA synthesis and RNA controls and produces protein synthesis is called gene expression. Okay. DNA itself is the genetic molecule that organisms inherit from their parents. Many long DNA strands, we refer to it as a chromosome. They can carry, more than 1,000 genes, depending on the organism. This could vary. Cells reproduce, they're passing copies of this DNA strand from parent to offspring. So we're going to see this whole process of mitosis and meiosis later on. right? They copy the DNA. Each new DNA strand, it will be passed down to each new cell. DNA itself is not sufficient. So this is very important. Even though that DNA carries all the information, it alone cannot execute molecular interactions, right? The protein must carry out a certain action. Okay, so the DNA has the information to create the protein makes the action, like hemoglobin and oxygen transport. All right, so RNA serves, as we're going to see later on, as an intermediate relay messenger, like a little recipe in a cookbook, right? It'll take the information from DNA in the form of mRNA, referred to as messenger RNA. It moves on to the ribosome. The ribosome will read this RNA and this sequence of RNA to produce a particular strand of amino acids, ultimately a polypeptide. Okay, as you can see here, DNA being for eukaryotic gene expression, you have DNA in the nucleus, right? This is referred to as transcription. It will produce an mRNA strand. Of course, there's more processes like splicing and pre-mRNA and mature mRNA. But ultimately, the RNA will leave the nucleus through the nuclear pores, so the ribosome can come in and read it to produce a polypeptide. Ultimately, that is gene expression. Okay. So of course, you have that ribosome in the cytoplasm. DNA stays in the nucleus. Components of nucleic acids. This is more particular of what you need to know for now, right? Nucleic acids, they could also be referred to as polynucleotides. But the nucleotide itself, what is it composed of? So you have this kind of little list here composed of 3 parts. This is something you must know as a biologist, right? 5-carbon sugar, a pentose, a nitrogenous base. Of course, it contains nitrogen, and 1 to 3 phosphate groups, right? And 2 of these phosphates are actually lost during the polymerization process. If you can see here, this is how a nucleotide looks like. You have your sugar, you have the nitrogenous base, and you have the phosphate group. Okay, each nucleotide differs in their nitrogenous base, unless you have 2 identical ones in the sequence. Okay, when you have a nucleotide and it is without a phosphate group, it is called a nucleoside. So you have the nitrogenous base and the sugar isolated. Okay, but when they're in a chain, you can see here, this is a polynucleotide. All right, and they can chain up covalently, where you have this sugar, phosphate, sugar, phosphate, sugar, phosphate, and the nitrogenous base is RNA particular orientation there. In itself, though, that is what a nucleotide is. Okay, this is the monomer. There are different types of monomers, so we are going to take a look at. Okay, so there's different types of nucleotides, depending on the nitrogenous base that they have. Okay, each nitrogenous base can have 1 or 2 rings, and they're grouped into 2 groups here, into 2 families. They're referred to as bases, and you have the pyrimidines or pyrimidines, and you have the purines. Okay, so you can see them here. The pyrimidines, the pyrimidines is cytosine, thymine, and uracil. Uracil used for RNA, thymine you use for DNA. And then you have the 2-ringed groups, the adenine and guanine. Okay, so you have these types of nitrogenous bases. Again, like I mentioned before, thymine is used to produce DNA. Uracil is used to produce RNA. Both molecules use the same guanine, cytosine, and adenine. They're abbreviated by the first letter. By the first letter of their names, right? C, T, U, A, and G. And in terms of sugars, right? They're pentose sugars, but DNA is referred to as deoxyribose. Make sure you use this level of specificity when you're writing, in any test, right? And RNA is ribose. So deoxyribonucleic acid comes from the deoxyribose here. The difference is that deoxyribose lacks oxygen at the second carbon. Okay, so if you take a look here, there's a lack of oxygen here for DNA, and there's an oxygen for RNA in ribose. Okay, just a little bit continuing there. Nucleotide polymers, right? So a nucleotide linkage essentially undergoes condensation reactions. Adjacent nucleotides, they're joined by phosphodiester linkage covalently. All right, a phosphate group links two sugars. A phosphate group links two sugars, so that creates a sugar phosphate backbone, as I showed you in the previous picture there, and we're going to come back to that. Okay, so the phosphate end has a 5' carbon, and the hydroxyl group of the sugar has a 3' carbon. So due to this, DNA has directionality, like one-way streets. So there's a 5' end and there's a 3' end. Okay, the sequence of bases is unique to each gene and provides specific information. We're going to take a look at this in a lot more detail. Sequence will specify the protein 3D structure. Okay, let me see if I have a picture here. I'm sure I do. Okay, so if you see here one strand here, you have your sugar end and you have your phosphate end here. I might go to the previous slide. Okay, this is much better. So here you have the 5' end, basically the sugar, the 3' carbon. Sorry about the bell there. Then you have your 5' carbon, which links to that oxygen of the phosphate group. Okay, so you have an orientation, directionality, 3', 5' end. Okay, nucleotides have directionality. Very important when we get into replication and gene expression. For now, that's all you need to know. Okay, continuing to the structure of DNA and RNA molecules. So DNA has two polynucleotides or strands that wind around each other. So they form a double helix. As you can see here in this figure, right? One strand is wound around another strand, but they run in opposite directions because of the nature of the 3' to 5' direction. In their one-way streets, in order for them to actually bind together, they have to be going in opposite directions. Okay, and that is referred to as being anti-parallel. The sugar phosphate backbones, they're on the outside of the helix. Okay, so have that in mind, that term, right? The sugar phosphate backbone. And the nitrogenous bases, for both pairs, they're faced inward, and they bind together complementary, right? So that means that adenine will always bind to thymine, guanine will always bind with cytosine through hydrogen bonding. Okay, the pairing is held, again, hydrogen bonds. Most DNA molecules are incredibly long, right? 1,000 to million base pairs. So yes, remember that each individual hydrogen bond is relatively weak, but when you have all of these nitrogen bonds working together, it tends to be incredibly stable. Okay, we're going to take a look more in detail when we get into DNA. Base pairing only occurs, and the name tells you, base pairing occurs in very strict pairs, right? So you have adenine with thymine, cytosine with guanine. It's complementary, or it's said to be complementary. So this idea enables this property, moreover, it enables DNA to copy itself during cell division, or any time that the DNA needs to, or the cell needs to replicate, right? And you want to have more daughter cells, and you're going to have identical copies of DNA from a parent cell. Again, this is another topic we're going to see more in detail. It's called DNA replication. RNA, talking a little bit about RNA, it's a molecule that exists as single strands, and it is complementary base pairing can still occur, between different regions of the molecule that the base pairs, become complementary to each other, right? So an example of this is transfer RNA or tRNA, right? This is a one type of RNA molecule that brings and transports amino acid molecules to the ribosome during translation of protein synthesis process. Okay, so this is an example of a tRNA. So this is one single strand that you have regions that are complementary. So if you see here, this is an example of a base pair joining G with C, G with C, U with A, right? So even though it's a single strand, again, you can have regions of complementary base pairing. Okay, there are different types of RNA, and they can vary in shape and function, right? They're very versatile molecules, and biologists do think that RNA came first during evolution due to this idea that it was a single strand before DNA came in as a more stable molecule and a more as a carrier of information there. Okay. And the last part that I want to talk about for this chapter is 5.6. Genomics and proteomics have transformed biological inquiry and applications. Okay, so the first half of the 20th century, you get a role of DNA being established, right? The whole concept of inheritance. In 1953, the structure of DNA was described, and it was understood to code for, specific amino acid sequences, right? So scientists sought to decode these genes by understanding the nucleotide sequences, essentially studying base sequence. So at that time, sequencing techniques, of course, wasn't the best. They were limited with the technology of the time, right? So, but nevertheless, they were able to discover sequences of different organisms and of particular genes. More sequencing led to more questions. How does gene regulation work? What does this particular protein do that is being created from this particular sequence? So discovering genomes was important to fully understand the relationship between proteins and DNA. So in the 1980s, scientists launched a project, a very massive undertaking, as I'm sure you've heard, the Human Genome Project, which aimed to, sequence 3 billion bases, which was finished around the 2000s, right? At that time, since that time, DNA technology and sequencing technology has greatly improved, right? Sequencing became faster. It became cheaper, more affordable for individual labs to be doing it at a regular basis, right? Many genomes since then have been created, annotated, established in databases, right? So you begin to have this whole kind of paradigm of bioinformatics, which is computational software and tools to help analyze large data sets, sequencing data, to help make sense of all of this raw data that we're collecting. Okay, so you get genomics. Genomics is an approach to analyze data sets of different species and the genomes of a single species, compare them, right? Understand how they work. And you get proteomics. Proteomics, similarly, you analyze protein structure, amino acid sequencing, and how specific sequences lead to particular structures and particular functions. And one of the most significant impact of genomics and proteomics is the contribution to understanding evolution, degrees of relatedness at the sequence level, at the base pair level, at the amino acid level. So scientists were able to, correlate sequence similarity to degrees of relatedness, right? Very, very powerful tools and methods to understand, evolution. So DNA and proteins are also used as tape measures of evolution. So shared traits like hair and milk production in mammals indicate a certain level of shared ancestry. DNA carries heritable information in genes. These genes can document, right, an organism's hereditary background. And the more similar these genes are for particular proteins, the more likely they're evolutionarily similar or more closely related, right? So nucleotide sequences in DNA are passed down again from parent to offspring and determine protein amino acid sequences. So siblings would share more DNA and protein than unrelated individuals of the same species. So you can get a sense of how closely related they are. Okay. Evolutionary relationships, they can be traced through molecular genealogy, comparing, again, DNA and protein sequences. As I mentioned before, closely related species have more similar DNA than distant related species. An example is the beta polypeptide chain of human hemoglobin, which differs only by one amino acid from gorillas, but 67 amino acids from frogs. So definitely you can see a correlation there. Whole genome comparisons show 95 to 98% of their genomes with chimpanzees, but only 85% with mice, comparing humans, with chimpanzees. Okay. So molecular biology provides a very valuable tool for assessing evolutionary relationships. Okay. Again, here you have a concept check. And with this, we essentially finish chapter 5. I want to just kind of slide through and for you to see making connections, contributions of genomics and proteomics and biology. Definitely you see a lot of insight in paleontology and medical science and in evolution, even species interaction and conservation biology. Okay, let me go to the second panel for you to see. You're free to pause the video if you like. To use scientific skills exercises so you can see some examples of practical applications. Okay, problem solving exercises. Okay, moving on to the chapter review. So these are summary of key concepts. I'll just kind of slide through here so you can get a sense. You can pause the video if you like. And some chapter review questions that you can try to tackle there. Okay, so with that, we finished the video. Thank you for watching, and I hope you enjoyed. I hope it was very insightful, and I'll be back with another one. Thank you.